Hi Nat5, so we're looking at key area 5 in cell biology, and this is genetic engineering. So this has to be the big question, why would you use genetic engineering? Because we have ways of trying to get what we want, and we've done that for generations. What you need to do is you just select parents that show what you want, and then you keep on doing that and you get what you need at the end of it. But if we look at something like a dog, like the ancestral dog, and all the many different breeds that we now have, they've all been a selection for different things. And then that selection has been fixed because we picked the parents. And that works really well, but it does take quite a long time because you have to go by their generation time, and you can only use what's already in the genetics. So selective breeding problems, which are the ones we just mentioned, and also there are other issues which can kick in, including something called inbreeding depression, higher if we're looking at that. Okay, so we need to find other ways, and one of them is to use genetic engineering. So genetic engineering is taking genetic material from one cell and putting it into another cell. That sounds really simple, it's really not. Okay, the organism you've now got we call a genetically modified organism, which is GM for genetically modified, or GMO for genetically modified organism. Okay, and if the gene came from a different species, which normally it has, then we can also say it's transgenic because we've moved the gene from somewhere else. Okay, and this is not how it works at all. So we need to have some ideas of how we started working on this, and so one of the things that they started using was E. coli because it's really common, it grows really easily, you can look after it really well, and it has a couple of really important things about it. So its size is not a big deal. Normal things about bacteria, it's got a cell wall, a cell membrane, a cytoplasm, and it has this single circular chromosome. But if you remember when we're doing cell structure, we have an extra few things which you have inside some bacteria, and these are plasmids. And plasmids are really important for this. I tend to think of it as that single circular chromosome is like the operating system in your phone. You don't mess with that, because if you mess with that the phone stops working. But the plasmids are apps. So you can add apps, you can edit apps, you can put new functionality in by changing the apps. And that's what plasmids do, they are extra bits. So these extra bits, the cell doesn't need them, so they could live without them. But it also means we could take them out, that'll kill the cell that we did that to, alter it, put it into new cells, and now you've got new DNA. So we can take our plasmid, literally slice it open, and it creates something called a sticky end, and then we can take a gene from somewhere else entirely different. So this one's a long chromosome, so say this was from a human, take out the gene that we wanted, also with its sticky ends, and stick them together, and we get our modified plasmid, and this plasmid is now carrying DNA which does what we want it to do. So here are our stages. So you take the gene you want, you cut it out, you find your plasmid, you cut it open, you stick in the gene, and then you put it back into a new cell. That cell then multiplies, and once it's multiplied it will produce whatever you want it to produce with the DNA you've given it. So one of the first things that we've done with this, or one of the really big things we've done with this, is to produce human insulin. Because human insulin is what you need if you are insulin dependent diabetic, but you can't get human insulin from humans. You would need to smash up lots and lots of pancreas, and we're not going to do that. So what we used to use was pig insulin or cow insulin. So that's not an option in the long term because it starts to actually react badly to your system. So what you want is human insulin, and this is how we now get it. So there are bacteria and there are also yeast which have had your plasmids taken, cut, stuck in with an insulin gene, and then engineered. So there are two enzymes that you need to know for this. So when I said cut, obviously that has to happen somehow, and the cutting and sticking is using enzymes, which shouldn't surprise you because they do all the reactions that you have inside cells. So finding an enzyme to do this, a major step forward in genetic engineering, and you will have people who will actually argue this is one of the biggest discoveries that's been made in science. So what we have are restriction endonucleases, which are particular enzymes that snap and cut DNA, and then you've got ligase, which sticks it together. You just need to know the names. So your cutting ones are restriction endonucleases, and your ligases join. Okay, so there's a huge range of GMOs out there, and we've already mentioned the human insulin ones, but we do have also lots to do with crops, because food production is hugely important. So things like being resistant to herbicides means that you can spray the whole crop and the crop's okay, but the rest of the weeds that are killed. You can have things that are less likely to be destroyed by pests, and you can even enhance things so they have more nutrition. Golden rice has a particular vitamin enrichment. So these are all examples that it would be worth you knowing at least a few. The human insulin one is the one I expect you to know, and you need to know the stages. And that is everything for genetic engineering.